In this video, we will talk about the calcium and phosphate regulation. We often think that phosphate is excreted out in response to parathyroid hormone. Well, this is true, and in many books we can find this statement that the action of PTH on kidneys is excretion of phosphate. But technically speaking, this statement is not telling the whole truth. It is hideous and thus slightly misleading. This statement that PTH causes excretion of phosphate gives us a false feeling as if tubules actively secrete out phosphate via pumps into the lumen. But as you can see, this is wrong. What PTH actually does is it prevents the reabsorption of phosphate and thus causes phosphate excretion or phosphaturia. So in this video, we will also clear out this myth. Apart from this, we will discuss the calcium profile in various endocrine disorders of parathyroid gland and in some of the metabolic bone disorders as well. And finally, we will construct this table. Instead of remembering that PTH hormone causes phosphate excretion as given in many books and sources, I ask you to rather remember it as PTH hormone prevents phosphate reabsorption. And you must be wondering, why is this point so important? It is important because we get such questions. Here, both option C and option D are correct, but option D is the best answer. And if you had remembered the bookish statement that PTH hormone causes phosphate excretion, then you would have marked option C, which would have been incorrect. Although, if PTH hormone prevents phosphate reabsorption, it still ultimately leads to phosphate excretion in urine. But the actual mechanism is that it prevents phosphate reabsorption, and that is why option D is the answer here. So you can see how MCQs are interplay of words. Statement 1 is false because we know that PTH causes increased vitamin D formation in the kidneys. It is one of the major stimulators of vitamin D synthesis in the kidneys. Similarly, statement 2 is false because we know that PTH causes increased reabsorption of calcium from the kidneys. So statement 1 and statement 2 can be easily ruled out. The only confusion is between statement 3 and statement 4. But as previously said, statement 4 is the best answer. But even better answer than statement 4, or you can say the bestest answer, is that PTH decreases phosphate reabsorption in the kidneys, leading to increased phosphate excretion in urine called as phosphaturia, thus leading to decreased phosphate levels in the plasma. Next, let's look at the actual mechanism of phosphate reabsorption in the kidneys. Phosphate reabsorption mostly occurs in the proximal tubules of the kidneys, aka PCT. Normally, in the PCT of kidneys, the tendency of body is to keep reabsorbing phosphate via the sodium phosphate cotransporter, also called as the NPT2A or NPT2C transporters. So you can remember it as like sodium is required at all times in the body, to it phosphate is also coupled. So there is always a basal reabsorption of phosphate taking place at all times in the body. This basal reabsorption of phosphate can be altered by hormones and factors, which in turn are secreted in response to body requirements of calcium and phosphate. Now, let's see what happens to this basal reabsorption of phosphate in the presence of parathyroid hormone. Parathyroid hormone comes via the blood and attaches on the parathyroid receptor denoted by R here on the proximal tubular cell. This leads to activation of the parathyroid receptor, which via the stimulatory G protein activates adenyl cyclase denoted by AC. Activation of adenyl cyclase leads to increased formation of cyclic AMP, which further activates the protein kinases. Protein kinases cause phosphorylation of NPT2A and NPT2C transporters. Phosphorylation of NPT2A and NPT2C transporters leads to their inhibition. If these transporters are inhibited, then that means phosphate reabsorption is also inhibited, along with inhibition of sodium reabsorption. Thus, phosphate is excreted out in the urine, leading to phosphaturia. Thus, we can see that PTH hormone prevents phosphate reabsorption, thus leading to increased phosphate excretion in urine and decreased serum phosphate levels. Remembering the whole downstream pathways, that is the stimulatory G protein or GS, the adenyl cyclase, the cyclic AMP, the protein kinases, is not at all important. The important thing to remember is that PTH acts on PTH receptor, stimulating it to cause inhibition of the sodium phosphate cotransporter. This much is sufficient. Next, let's look at the effect of vitamin D on phosphate reabsorption. Here, we can see that the proximal tubular cells have sodium phosphate cotransporters, namely NPT2A and NPT2C. These transporters are responsible for the basal reabsorption of phosphate. Proximal tubular cells or PCT are also the site for vitamin D activation. Therefore, we can see that 1-alpha hydroxylase is present here in the PCT. PCT is not only just a site for vitamin D activation, it is also a site for vitamin D action. Therefore, we can see the machinery responsible for vitamin D action in the PCT, like the vitamin D receptor and the vitamin D response elements, aka VDRE, on the DNA in the nucleus of proximal tubular cells. Vitamin D acts via a nuclear mechanism, but its receptors are cytoplasmic. We can also see the vitamin D binding protein, aka DBP. The final step in the activation of vitamin D takes place in the PCT. Half the work of activation of vitamin D, that is 25-hydroxylation, has already occurred in the liver. So in the liver, vitamin D is converted into 25-hydroxycholecalciferol. This 25-hydroxycholecalciferol is carried by vitamin D binding protein, aka DBP, to the site where final step in the activation of vitamin D occurs, that is the PCT. When this complex of vitamin D binding protein and 25-hydroxycholecalciferol reaches the PCT, they dissociate and 25-hydroxycholecalciferol enters the proximal tubular cell. where 1 alpha hydroxylase converts 25 hydroxycholecalciferol into 125 dihydroxycholecalciferol, aka calcitriol. Calcitriol is the fully activated form of vitamin D. Calcitriol next binds to the vitamin D receptor and both form a complex. It's not that the other vitamin D metabolites like 25-hydroxycholecalciferol cannot bind at the vitamin D receptor. They too can bind at vitamin D receptor. It's just that the affinity of vitamin D receptor is 1,000 times greater for calcitriol than any other vitamin D metabolite. And that is why calcitriol is given the title of the fully activated form of vitamin D. The complex of vitamin D and vitamin D receptor forms nuclear heterodimers with RXR. So next, a trio is created which consists of the fully active vitamin D receptor aka VDR and RXR. This trio then moves to the nucleus and interacts with the vitamin D responsive elements. These vitamin D responsive elements aka VDRE can upregulate or downregulate the expression of target genes to produce physiological actions. So here in renal phosphate reabsorption, calcitriol via the VDRE increases the expression of NPT2A transporters on the luminal side, therefore increasing phosphate reabsorption. In simpler terms, what this means is that the action of vitamin D on PCT and action of vitamin D on phosphate reabsorption is to increase the renal reabsorption of phosphate. And how is this possible? This is possible because VDRE are stimulated. As a result, there is increased transcription of genes responsible for the synthesis of NPT2A transporters. Consequently, there is increased translation. As a result, there is increased expression of NPT2A transporters on the luminal side. Therefore, there is increased phosphate reabsorption. This is what is meant by increased gene expression of sodium phosphate cotransporter subtype 2A, aka NPT2A transporter. Finally, vitamin D is cleared off and the proximal tubular cell attains its original state. Now, let's talk about the renal reabsorption of calcium. Maximum amount of filtered calcium is reabsorbed in the PCT. And this reabsorption of calcium in the PCT is not dependent on the hormones. It is occurring at all times in the body, and that is why this is the basal reabsorption of calcium. Only 5 to 10% of calcium reabsorption occurs in the DCT. And this reabsorption of calcium in DCT, this is hormone dependent. So the only factor that can affect this basal reabsorption of calcium in the PCT is the body requirement of calcium. Because if body requirements are high, then that means that the serum calcium levels are very low. And hence, this will increase this basal reabsorption of calcium in the PCT. Similarly, if body requirements of calcium are low, then this means that there is enough calcium in the serum. As a result of this, the basal reabsorption of calcium is decreased. Note in this image that the reabsorption of calcium is not across the proximal tubular cells. Rather, it is across the gaps that are present between the two adjacent proximal tubular cells. Since this basal reabsorption of calcium in the PCT occurs along the sides of the proximal tubular cells, that is why this is also called as the paracellular transport of calcium. You will be surprised to know that this paracellular transport of calcium does not require any transporters. So this is a passive process. Next, let's look at what happens to calcium reabsorption in the presence of vitamin D and PTH. The effect of both of these hormones is same, that is to increase the renal reabsorption of calcium. As already discussed, these hormones will act at DCT. But remember that maximum reabsorption of calcium occurs in the PCT. And this is hormone independent. The mechanism by which these hormones act on the DCT to increase the renal reabsorption of calcium revolves around the TRPV5 and TRPV6 transporters. So either the expression of these transporters is increased or their open state is stabilized by these hormones. Thus, ultimately, there is increased calcium entry into the distal tubular cells. So, this is what is happening normally in the distal tubular cells. Calcium comes into the distal tubular cells via the TRPV5 and TRPV6 transporters and leaves the cell into the blood via the calcium pump, aka plasma membrane calcium ATPase subtype 4, aka PMCA4, and NCX, aka sodium calcium exchanger. Now let's see the detailed mechanism of action of vitamin D. We can see the vitamin D receptor, the already activated form of vitamin D in the PCT, that is calcitriol, enters the distal tubular cells and attaches to these vitamin D receptors. This attachment stimulates the TRPV5 and TRPV6 transporters as well as calcium pumps. The open state of TRPV5 and TRPV6 transporters is stabilized by this attachment. As a result, there is increased calcium entry into the distal tubular cells. This attachment also stimulates the nuclear mechanism, leading to increased synthesis of TRPV5 and TRPV6 transporters. As a result, there is increased expression of TRPV5 and TRPV6 transporters on the luminal side of the distal tubular cells. Thus, calcium reabsorption is increased multifolds. The calcium that enters the distal tubular cells binds to calbindin and both form a complex. The activated VDR2 facilitates the formation of this complex. This complex then stimulates the calcium transporters present on the basal side, like the calcium pump and the NCX. And this effect is very logical because a lot of calcium is entering from the lumen of urinary tubules into the distal tubular cells. Therefore, these basal pumps like the calcium pump and NCX need to be hyperactive to transport this excess calcium out of the distal tubular cells into the blood. Otherwise, the calcium concentration in the distal tubular cells would be so high that the reabsorption of calcium will be stopped. So with the help of these basolateral calcium transporters, the calcium is thrown into the blood. Finally, vitamin D is cleared off and the distal tubular cells return to their original state. Let us also briefly look at the detailed mechanism of action of parathyroid hormone on calcium reabsorption in the DCT. This is what is happening normally in the DCT when there is no parathyroid hormone present. Hormone dependent calcium reabsorption occurs in the DCT. Maximum calcium reabsorption occurs in the PCT. This is hormone independent, which means that this reabsorption is occurring at all times in the body. Whether there be low calcium or high calcium, it does not matter. This reabsorption keeps on going. This fact has another major implication. That is, if body needs more calcium, then it has to increase this hormone mediated calcium reabsorption. And the only place where this occurs is the DCT, because hormones can act only at DCT to increase calcium reabsorption. And as already discussed, in PCT, calcium reabsorption is a passive paracellular transport process and not mediated by hormones. In contrast, phosphate reabsorption occurs in the PCT and hormones are involved. Thus, parathyroid hormone is elevated in such diseased states like hypocalcemia. This increased PTH hormone secretion leads to increased hormone dependent calcium reabsorption in the DCT. So, for calcium reabsorption in times of increased calcium requirements, the major compensatory mechanisms are the hormones, and these act on the DCT. So, major compensatory mechanisms occur in the DCT in times of increased calcium requirements. PCT cannot manage much. PCT can manage only mild levels of hypocalcemia. With this much information, let's continue further with the story of mechanism of action. Now the parathyroid hormone comes in and attaches at the parathyroid receptor on the distal tubular cells. The parathyroid receptor gets activated, and the same downstream machinery that we saw in phosphate reabsorption in the PCT is brought into action by the activated parathyroid hormone receptor. Thus, adenyl cyclase causes phosphorylation of the TRPV5 and TRPV6 transporters. The only difference here from the phosphate reabsorption is that in calcium reabsorption, the phosphorylation of TRPV5 and TRPV6 transporters stimulates them, whereas in phosphate reabsorption, phosphorylation of the NPT2A and NPT2C transporters inhibits them. And like in vitamin D, we saw that there is stabilization of the open state of TRPV5 and TRPV6 transporters. Similarly, here too, phosphorylation of TRPV5 and TRPV6 transporters by the downstream machinery of activated parathyroid hormone receptor causes stabilization of the open state of these transporters. Thus, there is increased calcium entry into the distal tubular cells. Other effects of PTH hormone is mediated via the activated form of vitamin D. Parathyroid hormone in the PCT causes increased activation of vitamin D into calcitriol. Calcitriol is then transported via the blood to the DCT, where it acts to increase the calcium reabsorption. We have already seen the effects of calcitriol on DCT. These effects are mediated via the TRPV5 and TRPV6 transporters. Same two mechanisms operate here too. That is, stabilization of the open state of the TRPV5 and TRPV6 transporters, and increased synthesis of TRPV5 and TRPV6 transporters, leading to increased expression of these transporters on the luminal side. Both these effects are responsible for the increased reabsorption of calcium in the DCT. Calcitriol also increases the activity of PMCA4 and NCX, which throw out this increasingly reabsorbed calcium into the blood. So we can say that most of the effects of parathyroid hormone on calcium reabsorption are mediated via calcitriol, because in PCT, PTH hormone acts to increase the activation of vitamin D into calcitriol. We have finally come to an end of this part of the video. Let's just quickly summarize all the important points discussed in this part of the video. The final step in the activation of vitamin D occurs in the PCT and is carried out by 1-alpha hydroxylase. This final step in the activation of vitamin D is also called as 1-alpha hydroxylation of vitamin D. Some part of the actions of PTH hormone is carried out by vitamin D, as PTH hormone increases the activation of vitamin D. Vitamin D increases the levels of both, that is, phosphate and calcium in the blood. But PTH hormone has opposite actions on calcium and phosphate levels. It increases serum calcium levels but decreases serum phosphate levels. Statement 1. Maximum phosphate reabsorption occurs in the PCT. This is the basal reabsorption of phosphate and is hormone dependent. Since this reabsorption is hormone dependent, therefore statement 2 is that both vitamin D and PTH hormone have to act on PCT for their effects on phosphate reabsorption. Statement 3. Like phosphate reabsorption, maximum calcium reabsorption too occurs in the PCT. Another similarity is that this too is the basal reabsorption of calcium, like we saw in phosphate reabsorption. The difference is that this calcium reabsorption is not hormone dependent. Since this calcium reabsorption is not hormone dependent, therefore statement 4 is that for PTH hormone and vitamin D to show their respective actions on calcium reabsorption, they have to act on DCT. Thus, hormone dependent phosphate reabsorption occurs in the PCT, whereas hormone dependent calcium reabsorption occurs in the DCT.